Good morning and welcome to the next series of Bio 110 lectures. Today we're going to be looking at the cytoskeleton. Today's lecture will concern one member of the cytoskeleton family, the actin filaments. Here in this slide I'm going to bring up the objectives that chapter 17 has. There you go. So please take a time to look at the objectives. Again, I'm not going to discuss them directly here, but make sure that when you study, you master the objectives shown here in the slide. Now, when we think about the cytoskeleton, we are thinking about structural proteins that serve the cell's purpose of maintaining shape, working during different biological properties, as well as giving the cell strength. So we have the actin filaments, we have microtubules, and we have intermediate filaments. This lecture today is going to be concerning actin filaments, shown here on the right. So now actin filaments have multiple functions in the cell. They could be used as structural support in the cell, they could be used for movement and motility, and they can be used during cell division. So it is very similar in some of intermediate filaments and microtubules. Some of them are going to have similar properties. But actin is one of the most abundant proteins present in the cell, and it's designed and used for multiple purposes during the life of the cell. The actin filament, it's made of independent monomers, the actin monomers. And the actin monomers are arranged together in a helical structure. This helical structure has a plus end, and this also has a minus end. We also call the minus L the pointed end and the plus end the barbed end. So as you can notice, the actin filament is a very long, strong structure. Actin monomers are ATP binding proteins, and they can either bind ATP or they can bind ADP. When they bind ATP, the actin monomers are able to assemble directly, and that helps with the polarization of the molecule. We call this the nucleation. When they hydrolyze ATP and release phosphate, the actin molecules have a little less affinity to stay together, and actin with bound ATP can depolarize at the minus end. The actin filament is assembled through a process that we call treadmilling. As you shown here in this image, an actin ADP monomer will bind, here shown in position 1, and when the next actin monomer binds again, that has shifted the position of the original monomer by one monomer down. So as more actin monomers containing ADP are binding, and they subsequently hydrolyze ATP into ADP and release a phosphate, the monomers move down the treadmill until they reach the end of the filament, where they can actually depolymerize and fall off. Eventually, the ADP on the actin bound with ADP can be exchanged for ATP, and the monomer pool can then be used to elongate the actin filament. This process is very dynamic, and the molecule of actin filament is constantly being generated by adding monomers at the end, at the plus end of the protein, and having the ADP monomers being released. So interestingly, the ATP bound actin polymerizes faster than the ADP minus actin, which can then fall off a little bit easier. In the cytoplasm of the cell, you have an actin monomer pool, and that actin monomer pool could be maintained as monomers when they are bound, when these ATP bound monomers are bound by monomer sequestering proteins. At the same time, the monomers could begin assembly into a filament, and that filament often requires the binding of a nucleating protein, and that nucleating protein will facilitate the assembly of the monomers into an actin filament. Now, the actin filaments themselves can interact with other proteins. Some of them, the severin proteins, can shorten the actin filaments, cross-linking proteins in the cell cortex, can help maintain actin filaments crisscross. You have capping proteins that can bind the plus end, so the monomer doesn't grow anymore. Or you can have side-binding proteins, we're going to look at some of this in the heart, that can associate with the helix of actin to move. We also have proteins like the myosin motors, which are going to use the actin filaments as threads in order to exert movement, or binding proteins, for example, in the filopodia that will allow the actin filaments to stay stick. So we're going to take a look at the cross-linking proteins in the cell cortex, and we're going to be looking at muscle cells to look at the myosin motor protein. Another one that we're going to take a more closer look is going to be the side-binding proteins. Now, as we discussed earlier, in red blood cells, you have spectrin that is helping maintain the structure of the red blood cell by being part of the network of molecules in the cell cortex of the red blood cell. So spectrin helps maintain that by forming a beautiful network within the cell. And actin is one of the proteins that could then bind and help maintain the spectrin pull. Now we have other proteins like myosin. Myosins are actin-dependent motor proteins, and they come in different forms. Actin-1, Myosin 1, it's the smaller chain of the actin family. It contains a structure that includes a motor. Myosin motors are ATP binding motors that could then use the actin track to be able to move. And the particular Myosin 1, has membrane binding activity, so it could be bound to the membrane or to endocytic vesicles, and then in that way, this little motor is used to then move those vesicles or the membrane along the actin tracks. One of the most common forms of myosin is Myosin II. Myosin II has a very long coil it's a coil-coil interaction, and it basically binds two different myosin heads together. This is the main myosin present in muscle that is going to allow for muscle contraction. The other versions of myosin, Myosin 5, Myosin 6, and Myosin 11, are different versions of the same structure. Some of them used for vesicular transport, endocytosis, and cytoplasmic streaming. In particular, let's take a look at Myosin II and how it's going to be used in muscle contraction. Now, Myosin II, as I mentioned, it involves two Myosin II monomers that are going to bind together and coil by a helix-coil-helix dimer. Now, this molecule is able then to associate together into a much larger filament that forms what we call the thick filament, or in the muscle. So as you can see here in the lower part, the Myosin II molecules, the dimers, can associate themselves like you're having flowers in a arrangement. So the bare region here in the middle, it's just the coil-coil dimer part of the myosin, and then just like a plant for a Brussels sprout, now the motor heads are arranged together in a structure where the thousands of myosin heads are distributed to the left or to the right. Now, this myosin coil dimers arrange themselves in filaments, and these are the filaments that are going to be used to exercise muscle movement. Now, myosin is going to convert the chemical energy of ATP to mechanical energy in contraction. So when we think about a myosin monomer, the Myosin II filament is going to attach itself to a filament and is going to mediate a contractile force. The myosin heads are they have a polarity. They like to move to the positive end of the plus end of the myosin filament, and therefore, when they bind to independent actin filaments, the myosins are going to move one towards the positive end and the other one towards the positive end. That is going to result in the movement of the filaments away from one another, shortening the distance between them. So now, how does the myosin enzyme work? So the myosin head, it has ATP binding capacity. And as you see over here in the top part of this figure, the myosin head, it's bound to an actin, the active filament to an actin monomer. So look at it over here, this one right here in the middle, that is a different slider color of orange. At this state, we have the myosin head firmly bound to actin in what we call the rigor state. That rigor state is a stable, rigid structure. And as you can notice as well, there is no ATP or ADP bound to that myosin head. Now, an ATP molecule will come in and it will bind to the myosin head, and that is going to induce the release of the myosin head from the actin filament. When ATP is hydrolyzed by myosin ATPase activity, that is now going to hydrolyze the ATP to ADP and phosphate, and both of them are going to remain attached to the myosin head. But that hydrolysis, as you can appreciate from the picture, has induced a slight conformational change in the myosin head, moving from the original position that it was to a little bit to the right in a different state. Now, the weak myosin binding to actin induces a conformational change, and that conformational change is going to release the phosphate, and that's going to allow the myosin head ADP to bind to a different part of the monomer. Now, releasing the phosphate induces a tight myosin binding to the actin filament, and that is going to now induce the power stroke. As the power stroke is induced, that's going to move the myosin, move the actin molecule down. That is going to induce the release of ADP, and you have now reset the system back at the rigor state. So that concludes one entire cycle. So the cycle of myosin binding to ATP, separating from the actin filament, hydrolysis of ATP into ADP and phosphate, as well as the induction of the weak binding to the monomer again, to the actin filament at a different monomer position, and then the induction of the power stroke and release of ADP gets repeated over and over again, allowing the myosin head to move towards the positive end of the actin filament. So now, because of all these events, the myosin head has moved towards the plus direction. To illustrate this better, at a bit more molecular level, I'm going to show you this following film.
Muscle myosin is a dimer with two identical motor heads that act independently. Each myosin head has a catalytic core and an attached lever arm. A coiled coil rod ties the two heads together and tethers them to the thick filament seen on top. The helical actin filament is shown at the bottom. In the beginning of the movie, the myosin heads contain bound ADP and phosphate and have weak affinity for actin. Once one of the heads docks properly onto an actin subunit, phosphate is released. Phosphate release strengthens the binding of the myosin head to actin and also triggers the force-generating power stroke that moves the actin filament. ADP then dissociates, and ATP binds to the empty nucleotide binding site, causing the myosin head to detach from the actin filament. On the detached head, ATP is hydrolyzed, which recocks the lever arm back to its pre-stroke state. Thus, like a spring, the arm stores the energy released by ATP hydrolysis, and the cycle can repeat. The actin filament does not slide back after being released by the motor head, because there are many other myosin molecules also attached to it, holding it under tension.
The swing of the lever arm can be directly observed on single myosin molecules, here visualized by high-speed atomic force microscopy.
So let's take a look at how the myosin and the actin work together to induce muscle contraction. In a muscle fiber, which is a muscle cell composed after the fusion of hundreds of independent muscle cells, we have a particular structure called the myofibril. The myofibril, it's the contracting element of the muscle cell. When we take a closer look at the myofibril, we see that the myofibril has distinct lines that can cross about it. And those lines are the images of the structure called a sarcomere. A sarcomere is the actual contractile unit within the myofibril. Now, the sarcomere is composed of actin and myosin filaments. We have what we call the thick filament. The thick filament is going to be the myosin filament in the middle. And the thin filament, it's the actin filament that is bound together in the sarcomere by a protein called the C disc. Now, when the muscle is induced to contract, the myosin thick filament is able to move and bind to actin and move towards the positive side, inducing a contraction of the sarcomere structure. As you can see, as I mentioned, the C disc have the actin cross-linking proteins that are keeping the sarcomere nicely organized and tethered in place. To illustrate this, we have to take a look at the muscle fibers themselves to understand how the muscle fiber is indicated to contract. So muscle fibers, as we have discussed earlier, contain T-tubules, which are invaginations of the plasma membrane that are going to go across the cytoplasm until they get to the myofibril region of the muscle fiber. As illustrated here in this area, you have also associated with the myofibril the sarcoplasmic reticulum, which is the equivalent of the endoplasmic reticulum in muscle cells. This is shown here in green. So as you see, the endoplasmic reticulum, the sarcoplasmic reticulum, and the T-tubule are in close association with one another. Here shown in this electron micrograph, you can see one of the small invaginations of the T-tubule and how that is very close together to the sarcoplasmic reticulum in a very beautifully organized structure. Go back and remember the membrane depolarization lectures, where we have a membrane potential that has been given by the neuron cell into the muscle cell. The muscle cell has received the signals for acetylcholine, let's say. An acetylcholine receptor has been bound by the acetylcholine, opening the channel, allowing then sodium ions to move in. That initiates a depolarization that is going to result in an action potential. That action potential will travel the membrane of the muscle cell, and when it gets into the T-tubule, the action potential will go inside the T-tubule, following the membrane until it reaches a voltage-gated calcium channel. In particular, this voltage-gated muscle channel, which is going to be in the area where the T-tubule is closely associated with the lumen, with the sarcoplasmic reticulum, it's going to induce the action potential that is coming down the T-tubule. It's going to induce a conformational change in the voltage-gated calcium channel that is going to release calcium into the cytoplasm of the muscle cell. But at the same token, that change in structure is going to open a calcium channel that is present on the sarcoplasmic reticulum. And that release of calcium, it's going to now inundate the cytoplasm of the muscle cell, and is going to result in the myofibril contraction. Now let's take a moment to look at how contraction in the muscle cell happens. Inside the sarcomere, as I mentioned to you, we have actin filaments that are spread as distinct filaments in the sarcomere. However, those actin filaments are going to be bound by proteins. Two of these proteins are important in muscle contraction. The first one of them is tropomyosin. Tropomyosin is a molecule that traverses the actin filament helix and goes around, as shown over here in the picture, sort of like the white or the red stripe around a candy cane, going around the entire helix, interacting directly with a particular position of the actin filament. Another molecule present in the thin filament is troponin. And troponin now is a calcium-sensitive complex of proteins that it's bound directly to tropomyosin. When calcium comes into the when calcium is released from the sarcoplasmic reticulum and invades the endoplasmic and invades the cytoplasm, now calcium is able to bind to the calcium troponin protein. That is going to induce a conformational change on the troponin that is going to be reflected in a conformational change in the tropomyosin molecule. Tropomyosin molecule, it's bound to the actin thin filament, but it's bound to the actin thin filament at the place where myosin will bind. So it is occluding the positions within the actin monomer that myosin would like to bind. So when calcium binds to troponin and troponin undergoes a conformational change and moves the tropomyosin molecule now away from the actin filament, it exposes the myosin binding site, shown here as these black dots. That now allows myosin, the thick filament, to bind to the actin thin filament and induce a contraction of the muscle. So as I mentioned, the troponin changes conformation, inducing a conformational change in the tropomyosin, and that is going to expose all the actin binding sites, the myosin actin binding sites on actin. So now here is another illustration of the same principle. So what we have again, in purple, it's the tropomyosin, and in green, blue, and light green and dark green and blue, we have the troponin complex. So when calcium comes into the system, that is going to induce the conformational change in troponin complex. And as you see over here from the side view, we have the troponin molecule in purple blocking the myosin binding sites on actin. So when calcium is present, the tropomyosin molecule is going to then be displaced slightly off, exposing the myosin binding sites, so now myosin can bind actin and mediate muscle contraction. So the troponin releases tropomyosin and exposes the binding sites to the myosin molecule, allowing for the sarcomeres to contract. When calcium goes away, the tropomyosin it's able then to move back and block the myosin binding sites on actin, and therefore induce muscle release. In this image from an article from the News of Physiological Science, I'm going to show you how that would look from the side. So here in figure A, we have the actin filament. In light gray, we have actin residues. And we have two actin residues that I want to point out to you. we have the lipo residues, and those are going to have electrostatic binding to myosin, but weakly. And we have the orange residues, and those are going to form very strong binding interaction with myosin. Now, let's bring tropomyosin into the picture. When we have tropomyosin into the picture, you can see that the orange residues, which are bound by myosin, are completely blocked. In this tropomyosin molecule, you can observe over here as a gauge to show you the movement, the arginine 90 amino acid in tropomyosin shown here in yellow, so you can actually see it on the side. On the tropomyosin here, it's shown as a dark gray molecule that crosses and blocks the orange residues, which are the ones which are having a really strong attachment to myosin. The lighter blue residues that have weaker attachments to myosin still can be seen, but not the orange ones. So when calcium is added, now you can start to see that the tropomyosin molecule begins to roll as a result of the troponin molecule having a conformational change. When the conformational change in troponin has been complete, you can now see that the arginine 90 residue in tropomyosin, instead of facing sideways, is now facing upwards. But that has now completely exposed the orange residues, which are the ones which myosin will bind very strongly. That allows myosin to bind to the actin filament tightly and therefore induce the contraction of the sarcomere. So now, remember, when we were having the discussion earlier in class, that neurotransmitter released by the neuron in the form of acetylcholine will bind to the acetylcholine receptor, which opens up a channel for sodium that induces a depolarization of the membrane, inducing an action potential. The action potential travels along the surface of the muscle by the opening of the voltage-gated sodium channel, and as you know, also the voltage-gated potassium channels are needed. And when the action potential travels down to the T-tubules, it's going to open up the voltage-gated calcium channels that eventually are going to open the calcium release channels in the sarcoplastic reticulum, allowing calcium to enter the muscle cell cytoplasm, and that results in muscle contraction. To show you this, I'm going to illustrate it on this movie.
When a neuron stimulates a muscle cell, an action potential sweeps over the plasma membrane of the muscle cell. The action potential releases internal stores of calcium that flow through the muscle cell and trigger a contraction. Muscle cells have an elaborate architecture that allows them to distribute calcium ions quickly throughout the cytosol. Deep tubular invaginations of the plasma membrane, called T-tubules, crisscross the cell. When the cell is stimulated, a wave of depolarization, that is, an action potential, spreads from the synapse over the plasma membrane and via the T-tubules deep into the cell. A voltage-sensitive protein in these membranes opens a calcium release channel in the adjacent sarcoplasmic reticulum, which is the major calcium store in muscle cells, thereby releasing a burst of calcium ions all throughout the cytosol of the cell. Within a contractile bundle of a muscle cell, called a myofibril, the calcium interacts with protein filaments to trigger contraction. In each contracting unit, or sarcomere, thin actin and thick myosin filaments are juxtaposed, but cannot interact in the absence of calcium. This is because myosin binding sites on the actin filaments are all covered by a rod-shaped protein called Tropomyosin. A calcium-sensitive complex, called Troponin, is attached to the end of each tropomyosin molecule. When calcium floods the cell, troponin binds to it, moving tropomyosin off the myosin binding sites. Opening the myosin binding site on the actin filaments allows the myosin motors to crawl along the actin, resulting in a contraction of the muscle fiber. Calcium is then quickly returned to the sarcoplasmic reticulum by the action of a calcium pump. Without calcium, myosin releases actin, and the filaments slide back to their original positions.
So now, for you to review, I have included a link over here. This link is from a thesis that was published online from a German university, and to get a better understanding of the cardiac muscle contraction biology, I will encourage you to read chapter 1. That will give you, it's a really good chapter, it's well written and has nice illustrations.